to day 8 of our summer research workshop on environmental sustainability held under the ANRF PAIR Network. As we explore new ways to protect our environment, we are very lucky to be joined by a leading expert in plant biotechnology. It is my pleasure to introduce our speaker today, Professor Mukesh Jain, from the School of Computational and Integrative Sciences at Jawaharlal Nehru University. Professor Jain began his academic journey by earning his MSc in biotechnology from Kurukshetra University and his PhD in plant molecular biology from the University of Delhi. Throughout his career, he has conducted pioneering research at esteemed institutions such as the National Institute of Plant Genome Research before joining as the faculty at JNU. His work is at the very forefront of modern science. Professor Jain focuses on plant biology, specifically in areas like functional genomics, chromatin biology, and epigenetics. His research into how plants handle stress and how seeds develop is helping us find creative, nature based ways to make agriculture more resilient. Professor Jain's work is recognized all over the world. He is a fellow of both the Indian National Science Academy and the National Academy of Agricultural Sciences. Beyond his many awards, he has made significant contribution to plant science through high impact research in journals like Science Advances and the Plant Journal. He has also created valuable tools such as the chickpea gene expression data atlas database, showing his deep commitment to sharing knowledge and helping researchers everywhere. We are truly grateful to have such an inspiring mentor guiding us today. His career shows us how careful scientific work can lead to real lasting solutions for a future. Ladies and gentlemen, please join me in a warm and heartfelt round of applause as we welcome to the stage a true visionary in plant science, Professor Mukesh Jain. Sir, we are so inspired to have you here to share wisdom. The floor is yours.
Thank you. I think I can reach. जरूरी है. Okay, good morning everyone. All of you are at the MSc level, MSc first year. Yeah? Okay. How many of you are with biology background? I guess most of you. Okay. Now I come to more specific questions other than biology background students. You are from which geology? Have you heard DNA sequencing? Read something about it? Okay. Say again. No. DNA is at the top level, then protein comes later. From DNA to RNA, and then from RNA to protein. So DNA is something which is called a genetic material, and it's part of every cell inside the nucleus, which defines what we are. It's how we will look. So it is also because of our DNA. I'm sure you must have at least these things heard about it. So point is that so all DNA is ATGC, right? Just 4 nucleotides. It's a combination of these ATGC which defines our whole DNA sequence, genetic material. But how to find out in what sequence, in what order these ATGC they are arranged? How big is the human genome? It's our genome. 3 billion bases, right? 3 GB. 3 GB is the human genome. So it's the arrangement of just 4 nucleotides in different ways that defines us, like what I am, how do I look, how do you look, or how do you look. So already Aradhana has introduced. So I work mainly in the areas of plant biology. But when we talk about DNA sequencing, it is same. So either it is a DNA from any other organism, or it is from plant, or it is for human like us. It is all ATGC. And in terms of sequencing also, it is same. So today I will be talking about some of the very latest methods of DNA sequencing, that how these methods have revolutionized the kind of research what we do in the areas of genomics or any DNA related research in terms of the downstream processes. So that's what the title of my talk is: next generation sequencing technologies. So there was first generation. I will introduce that also, especially for those who are not much aware about it. And then I will jump on to the next generation sequencing methods. And I have kept my talk very basic aspects of this. So just consider it is a primer that you know about what these technologies are, what we can do using these powerful technologies. And then later on, maybe at some point of time, you feel like that you have to implement or you can implement in some of your research, either in MSc or maybe after MSc, if you are going for further academic research like PhD, or even in industry, if you are going in this direction, so these can be implemented. So at least you will have some idea what the latest is going on in this field. Anyone otherwise have some idea? Means you have heard or read about next generation sequencing methods or technology? Anyone? No. But DNA sequencing, I guess all biology students, they know it. So coming to some history of DNA sequencing. We know 1953, the landmark when we had this DNA structure. And then it was in 1973 when the first ever any DNA nucleotide sequence was published. And this was the paper in PNAS, when they just sequenced 24 base nucleotide sequence. So it was a breakthrough at that point of time. So nowadays, 24 nucleotides, we just say we can just order to the company and they can synthesize it for us. But it was a breakthrough at that point of time. Yeah. Sorry, say again. Yeah, so it is something same time probably that. So you know that controversy also, right? So I'll not go into that controversy, right? So this is what the standard is. So history of DNA sequencing. So I'll just point it out here after the DNA model, which was basically proposed. It was in 1977. So is there any pointer also here? No. Okay. So 1977, when the first time these Sanger sequencing and Maxim Gilbert, so these two methods, what you mostly study in your textbooks, those were proposed. And at that time, if you will see at the left hand side, what was the efficiency of sequencing? The efficiency of sequencing was around 1000 bases per person per year. So in a year, one person can sequence around 1000 bases, 1 kilobase of the DNA sequence, because it used to be very cumbersome. A lot of manual work was required. But later on, of course, there were several developments. And in 1986, immediately after one of another breakthrough in molecular biology, what was the breakthrough just before 1986? Anybody remember? 1985, something happened. PCR, right? So immediately after that breakthrough, some automation in DNA sequencing came. So this was a partial automation, but still you can see on the left hand side, these are all very approximate numbers. Somebody may be very efficient also, some may be less efficient. So the sequencing efficiency was what, 25 kb, kilobase, 25,000 bases one person can sequence in 1 year. And then of course there were further improvements in terms of the machine, in terms of the chemistry, what I mean to say, the enzymes or other chemicals, buffers like you use in different reactions. So it was in 2002 when another breakthrough came, when actually the first next generation sequencing, what we will be talking about in today's lecture. So those came into picture. And here also you can see the efficiency of the sequencing. What is this number? 50 megabase per person per year. And then even further, there are a lot of improvements after this. Nowadays, we talk about around 6 terabytes of the data in just 1 week, or less than a week. 6 terabyte, you can imagine how much data it is. So, in my calculation, one scientist can sit throughout his life and can get maybe 30, 40 PhDs done, making sense of those 6 terabytes of the data in terms of ATGC. Right? Human genome is 3 GB. You remember it. And this we are talking about 6 terabytes. 6 terabytes meaning 6,000 GBs. So on the right hand side, there are two pictures are given. I don't know how many of you have seen. So the top picture is a manual method of DNA sequencing. When we used to have very big gels, you have seen SDS page. It's a very small. It used to be a very big page, and even casting of that gel sometimes used to take 3 to 4 days, because it's very thin, 1 mm, and there should not be any bubble. So it was approximately, what I say, maybe 1/6 of this, the board size. So that was even a challenge. And then there were radioactive labeling, then loading it on the gel, that was also in itself a challenge. And then once you have run the gel, then transferring that gel, so you have to do autoradiography. You have to transfer, you have to get all these bends on X-ray film. So even removing that big gel in one piece, so that was a challenge. So it used to take, even if you are perfect, so one reaction of around 100-150 bases, so you will take 1 week to 10 days. So the below figure is those who know about the DNA sequencing, maybe Sanger sequencing. This is the routine chromatogram. So this is how a DNA sequence looks like in a standard Sanger sequencing method nowadays. When you do one reaction, you send your sample to some company, so they will give you the sequence along with this chromatogram kind of thing. So you have certain peaks, you can see. Different colors of peaks are there, and different nucleotides are written on the top. So at what position, what nucleotide will be read, so that is written on the top of that. So this is how, again, just a small brush up. So what the DNA polymerase reaction happens, there is a phosphodiester bond formation between, means the two nucleotides when the chain grows, and then you have you add the chain terminators in Sanger sequencing, and your chain terminates. So this is a sum up of that Sanger sequencing, standard Sanger sequencing method. So you have different reactions. There are fluorophores labeled at each DDNTP. And when a DDNTP is added, your chain will terminate, and you will have the fragments of different size. And those, when you will load on the capillary, it's a kind of a, you can say, agar gel, like you see, on the basis of the size that you get different fragments. So in automated sequencers, there are capillaries, and capillaries are loaded with a certain kind of matrix. When your PCR mixture of these DNA fragments goes into that, so it is resolved according to its size, and you get this kind of hypothetical bending pattern. So this bending pattern is read by the machine. There are fluorescent cameras, so they read different kinds of fluorescence which is coming. So this is a digital image of that, what actually is generated. And now on the down, you can see this ATGC, so a chromatogram kind of, the real chromatogram you saw in the previous slide. So that is generated, and how the ATGC is called. And so this is a real chromatogram. So how it is read, the sequencing with the different colors of the fluorophores, and accordingly you have the order of ATGC. This is a routine Sanger sequencing, what you have read, or what you have learned in your textbooks. This is a machine. Just to give you an idea, if you have not seen a standard automated sequencer from ABI. Of course, there are different companies, they provide different kinds of sequencer. This is the most standard, and you can say in terms of Sanger is the most throughput machine. So, sorry, where you can run 96 reactions at a time. So there is a 96 well plate, and in each well you can run a sequencing reaction. So 96 samples can be sequenced. So here on this left hand side you can see that there are some waves going on. So these are actually the fluorescence which is coming from each of the well, so which is being read by the camera. So what you can do, I will come to this, why I am telling this, that why there is a need of next generation sequencing. So that will come into the next slide. So here we can do 96 reactions at the most in one run of Sanger sequencing, that is automated sequencer. And if we talk about as such as a whole, how the sequencing reaction happens in a Sanger method or automated sequencer, you have your target DNA, you do the PCR amplification of that, because maybe you have say whole genomic DNA, but a certain portion you would like to sequence, so you have to amplify that particular portion, and then you set up the sequencing reaction. You add all kinds of DD NTPs which are labeled with 4 different fluorophores along with the normal DNTPs, and PCR amplification will happen, and whenever there will be incorporation of DD-NTP, your reactions will stop and you will get the fragments of different size. And then these are loaded onto the machine. You have say capillary machine, and these are resolved, and finally you get the sequencing done in terms of the chromatogram. So now you just suppose, so few steps beyond this, before this also are done. So this is the step we actually send your DNA to a company for sequencing. So either you send as the in the form of PCR product, or you already have a fragment which is cloned in some vector, plasmid vector. I'm sure everyone knows plasmid vector. So plasmid vector have standard primer sites on the both ends like M13, SP6. So these kind of vectors, you PUC18, PUC19, very standard, PBR322, some of the vectors I am naming. So what company does then, they will use the standard primers like M13 primer, will amplify whatever has been cloned within the MCS, and that PCR product will be used. Or you send your PCR product and that is used in terms of the sequencing process. And this is how, again, the chromatogram, how it looks like, and how you measure the quality of the sequencing. Because why I am telling this, many of these things are related with the NGS also. We will again talk about these things when we will talk about NGS methods. So the peak of these individual, you can see the lines. So that actually defines indirectly as the quality of the base at that particular position. And that quality is measured in the form of a Fred score. Fred is the name of the scientist actually who gave the how you can calculate this score, and it is up to the scale of 0 to 40 in terms of the Sanger sequencing. 40 being highest, 0 being the lowest, and that basically based on the score you can calculate what is the probability of being an error at a particular position. So I will show that formula also, that how it is calculated. Like if there is a Fred score of 10, so it means there is a probability of 1 in 10. If it is 20, 1 in 100. If it is 30, it is 1 in 1000. If it is 40, 1 in 10,000. Because there is a PCR step, and PCR step you use Taq polymerase, and Taq polymerase has an error rate. Any DNA polymerase has an error rate. So there is always a chance that you may get an error. But what is the frequency of that error? So that can be defined by the quality score, which we are calling here as the Fred score. Now, for almost 40 years, Sanger method was the main method which was prevailing. Nowadays also we use, it's not like that it is outdated. Like for example, we have done cloning of a gene of interest, and then if you want to confirm its sequence is correct or is this the gene what you are interested in, still the Sanger method you use, because there is no point of using very high throughput method just if you want to have a sequence of a single gene, because that needs a lot of money also if you want to sequence at that throughput. So anyway, but if you want to go for the larger projects, of course, you need something very high throughput. And this, you know about the Human Genome Project. So it took almost 13 years to complete. Actually, this project initially was projected to be 20 years. So later on, it was speeded up and it took 13 years from 1990 to 2003. Now I ask you a question. You know about Sanger sequencing. In the current scenario, one reaction of the Sanger sequencing can generate 1000 bases, 1 kb in 1 hour when everything is ready. Means you have done PCR, you have cleaned it up, you have loaded it on the machine. The machine will take 1 hour. How much time do you think it will take to sequence one human genome? 3 GB, 3 billion bases, one reaction, 1 kb. And you can say 96 reactions can happen in one go. Let us take it in round of like 100 reactions. So 100 reactions in 1 hour. And each reaction is generating 1 kb. So how much data you are generating in 1 hour? 100 into 1000, right? So it is 10,000, 10 kb, right? Now you calculate how much time it will take to sequence a human genome, which is 3 GB. 3 GB is 3 billion bases. 3 into 10 raised to power 9. How many hours or how many days? I'm sure you are better than me in maths. This is a very important example just to realize where we are nowadays, means how much speed we have achieved. How many? 300,000 hours, right? So you can imagine how many years it will take. Now I am adding one more complexity. When you sequence it, there is always an error rate I mentioned for DNA polymerase. What is the error rate of DNA polymerase? 1 in around 700 bases. That's the standard, right? Even if you take 1 in 1000. So when you are sequencing, so you are incorporating so many errors. We don't want that many errors in a genome, because a single error, maybe one nucleotide difference is there, and that nucleotide is something very important for the functioning of a particular gene or something. So generally, when we used to do Sanger sequencing, we used to sequence on an average every nucleotide 10 times. So now whatever hours you have calculated, multiplied by 10. If you want to get a real DNA sequencing of a human genome. So even at that time when this project was launched, I don't remember the exact number. So there were around 100 labs, and there were thousands of sequencers which were working day and night, and thousands of manpower were working. So that was the aim. But later on, you must have heard about the Craig Venter name. Craig Venter is काफी famous in DNA sequencing. So he came up with some a new method of sequencing. Not exactly sequencing method, it's a strategy how you can sequence any genome. One thing is that you have 3 GB, you break it into the smaller fragments, say 150 kilobase or 200 kilobase, what we used to call as the, I will not go into those details. So the bacterial artificial chromosomes, you must have heard vectors. So that was one strategy, like you first break it into the bags and then you sequence individual bag. But he kind of came up with the whole genome sequence, shotgun sequencing method. So where you just chop up the whole genome into the small pieces of 1 kb or 1.5 kb and just sequence it random. Clone it into the vectors, just sequence it. We'll see it later on how to place it. So that was the strategy. And he could sequence the whole genome in 3 years. And that strategy, again, this public consortium, they also used it, and that's why their time was reduced from 20 years to 13 years. So now you realize that how much cumbersome was the Sanger sequencing method if you want to sequence the genome of any organism. Here is the example of the human genome. So initially, when the Sanger sequencing was there, each fragment was 500 bases, but nowadays we have methods you can go up to 1 kb. So that's what just now what I have discussed, that is there in the class in this slide actually, that how much time it will take to sequence an human genome. Now with time, the and also the cost factor, which I have not talked till now. So I am not sure how much per reaction nowadays is being charged for the Sanger sequencing. I have not got it done. But when I was doing PhD, it was around 600, 700 rupees per reaction. So now you multiply that, how much will be the cost of the genome sequencing, that to only sequencing part. And there is a lot of pre-processing. Even before reaching to the machine, you have to process the DNA a lot in several steps. So that cost is like other part. So now with the next generation sequencing, when the cost, you can see the how the scale is going down. So now it is still 2020 data. So you see the Moore's law goes by this, but the sequencing cost has kind of outperformed. So it is drastically down. So nowadays even we talk about $1,000 a single genome. So if you convert it, so within 1 lakh, you can do sequencing of a human genome. So here are some of the platforms. I will be talking very briefly about these platforms, about the next generation sequencing. The 454 sequencing, which was also called as the Roche, because the Roche was the company who basically came up with this method. There are a lot of reagents also in the lab, molecular biology reagents we use from this. And below this, the name of two different types of machines are mentioned. So no need to write this thing. I'll go one by one of some of the important aspects of this. Now the second point is that you need to remember is the Illumina. So which is the most prevailing sequencing method nowadays in the market. Earlier it was Solexa. When it started with the name Solexa. Later on it was taken over and now is the name Illumina. Then another method came by Applied Biosystems, who was leading the Sanger sequencing method. So they also came up with next generation sequencing method, and then with the solid platform. Then Iron Torrent, another platform came. Then Helicose, PacBio, Oxford Nanopore. So I'll talk about some of these methods which are very important nowadays if you really want to work in this direction. So before I go to the platforms, it's very simple workflow on the left hand side. You will see. You have the whole DNA at the top, any genomic DNA or any kind of DNA which you have isolated. You just do the fragmentation of this DNA. Fragmentation either you can do via some enzymes or it can be physical fragmentation like sonication. Sonication is more precise. Otherwise, earlier we used to do in our lab using the tips also. Like if you pass through a very fine tip multiple times, it gets sheared. But you still don't know. You cannot control what kind of fragments you will get. So better is the sonication method. So there you can control some of the parameters and actually you can make the DNA into smaller fragments, whatever you want. Then you get the fragments. But if you want to sequence, you need to have some known sequence. Otherwise, how you will start? You remember the DNA replication or even PCR, you need some primers. So you will get some DNA fragments, they are very small or some may be very large. For a good sequencing method or good sequencing output, we want to choose only a particular fragment size. So this is a size selection. You can see that all the fragments are of almost equal size. And here on the right hand side, it is shown that how a good fragment looks like. A DNA fragment means what we want to sequence, and along with that there are adapters, there are kind of primers added at the end. There is one more thing, barcode is also added. So barcode is something which you can identify. So that's in general what we call as a barcode. So why this barcode is added? Just I will correlate. I told you what is the throughput, 6 terabyte in certain platforms. So there are different kinds of machines which can generate different kinds of outputs. 6 terabyte, what I am telling is the maximum kind of throughput. So, but if you want to sequence it in such a platform, maybe you don't need that much of the data. It's all wastage. So what you can do is you can pool several samples, like the company people, they do it. You will get the samples from you, will get samples from you, from me, anybody. They will pool all the samples and will sequence in a single machine or in a single run. But then how do they separate out that data? This data belongs to you, this data belongs to me or somebody. So while sequencing, a barcode is added for each sample. So using that barcode is again, it's a nucleotide sequence. We are all talking about nucleotides only. It's a 6 to 8 or 10 nucleotide long unique sequence which is added. So using that barcode sequence, later on you can separate out that out of 6 TB, this 500 GB belongs to this person, this 500 GB or some TBs, whatever is the data requirement, this belongs to that person. So this lowers your cost to a very large extent, because within a single run they can run many samples, hundreds of samples, and then they can separate out the data for each sample and can cater to the requirement whatever is for individual researcher. So now I am coming to some of very generalized again what exactly is done when you actually do the sequencing. Emulsion word, I'm sure you have heard, it's a mix of oil and water. So this is actually more specific if we talk about the Roche platform, the first platform which I have told you. So what happens, you have a PCR mix and there is some emulsion oil is added. So I'm just again telling you some kind of bites of these methods. There are a lot of things to learn and you can just Google it out. You can watch very fine and beautiful videos are available. So they even you can catch it even more, I think, interactive way that how actually this happens. So just Google and then you can find YouTube videos. So a emulsion is formed. So this is a larger view of the emulsion. So there are beads into it. On the top right side, you can see the single bead. So this is one droplet. Within one droplet, there is one bead. And in one bead, there are DNA fragments attached around it. So one fragment is shown in a larger view. So this is the most ideal situation, that one droplet will have one bead and one DNA fragment. So you will have, of course, millions of such droplets and such beads. For each DNA fragment, it should be there within one bead. And then within that single droplet, PCR happens and you get this kind of cluster. The multiple copies of the same DNA fragment happen. So it's a kind of, you can say, mini bioreactor within that droplet, which is maybe of very small nanometers of size. And this PCR amplification happens, means this depends on what kind of technologies or chemistry you are using, but it's a very basic view of this, that how the PCR amplification happens and how it is read. This is another method which is used in Illumina, which happens in a solid support. So there it was in a solution, in some reaction mix, in liquid. But here it is happening on a solid support. So this is a slide on the right hand side. So on the left, first I have shown the same what you have seen the workflows. You have the DNA, you attach the adapters, you get the fragments. On the right hand side, there is already a flow cell where there are primers bind to it, which are corresponding to the adapter sequence, because adapters we are adding, so we already know the sequence. So corresponding to those primers are already there. So each fragment will go and bind to its corresponding primer sequence into it. And sorry, and there is a cluster generation. Again, the PCR amplification is happening on the solid surface. So there are thousands or millions of fragments are being amplified at the solid surface. So this is how again the PCR and the sequencing is happening. Again, just to give you some kind of visualization, this is how the machine looks like for the 454. The left hand side, it was a junior platform. Of course, the lesser output. Right hand side is the machine with the highest throughput. But I tell you now, this platform is obsolete. 2022, this went out of the market for several years. So it was kind of, I mean, very much in the market, and several projects were done. You just need to know some basics. Otherwise, any new data you cannot generate because nobody is providing services for this machine. So, but because the NGS history actually started from it, so it's better we need to know that this kind of platform existed, and at some point of time, if someone of you are interested in which is data already available in databases, maybe there is some data which is related or coming out from this platform and useful to you. So you can see some throughput, some figures are given here. The junior platform, where the read length was single sequence, which is the output of this machine, 400 bases, and up to 700 with the latest platform. And the total data output, you can see 35 megabases in 10 hours. And with this 700 bases, the bigger platform, it was 700 megabases with 1 million sequences. So still it is very, very much high throughput as compared to the Sanger method. And the accuracy is also 99.997%. That's what the company claims. So this is some views of that how the sequencing happens. So you have a picotiter plate. So you have the wells in those plates, beads which I have shown earlier. The emulsion PCR happens. This is how the beads goes and settles down in individual these wells which are of picometer size. This is how actually the plate looks like. So these are some microscopic views are shown. This is but actually how the flow cell looks like, where you load your PCR mixture. These are electron micrograph views of this. This is another platform, solid platform. This is how the machine looks like. Again, the throughput was here. You can see the read length is just 75 bases, smaller reads, but throughput is 180 GB with 3 million sequences per run, accuracy being 99.99%, and the runtime used to vary from 7 to 12 days depending on how much read length you want to get it. Again, this platform is also now obsolete, but now Illumina. So still, I think more than 90% of market is with the Illumina. So it's a very important method. So you can see there are different platforms available for this, and if you go by, it's a very evolving method. Every year they are coming up with some new platform, and they have different specifications for this. So if you go by this, the miniature model, which is IC 100, runtime is from 10 to 20 hours depending on how much read length you want, and it can generate 1.2 GB data with 4 million reads. And you see another NextSeq platform, so where up to 2 days and 360 GB of the data, and your reads could be 300 nucleotides in long, 2 into 300 nucleotides long. This was what I was talking about, the NovaSeq, where the actual data output is 6 TB, and then sometimes in NovaSeq X, multiple flow cells also you can use, and actually the output can increase to 16 terabytes per run. And there are different kinds of flow cells. Even 1 week I was telling, I think longer duration, they are just writing 2 days, 48 hours, 16 terabytes of the data if you run a machine in full strength, multiple flow cells. And read length is 150 bases. From the two ends, 150 from here, 150 from here. This is how the evolution of these different platforms happened in this particular Illumina method. So it's a very important technology. Anyone going into, or if you go to any kind of interviews and if you talk about NGS, so Illumina is kind of the first method people talk about. Now, another method, ion torrent. Again, this is also now off market. You will find machines available because the machine cost was low here. So, in many universities, even you can find, if maybe some people have bought it earlier. So, this is a personal genome machine and proton. So, some specifications of this machine are 200 base pair long read. But here again, different kinds of chips are available. So, if what kind of chip you are using, so your output can vary according to that. And your throughput can be 1 GB and even higher, again, what chip you are using within a time of around 5 hours. So, it's a kind of machine which was developed for metagenomics. I don't know, metagenomics, I am sure some of you must have heard. So, if you want to sequence the microbes, the DNA of the microbes, a combination of those, because they have the smaller genome, so this kind of machine can be very useful for that purpose. Now, in ion torrent, how actually sequencing happens? So, it is very sensitive. It's a semiconductor based. It can detect the change in pH up to the extent if there is a change in pH of just because of one hydrogen ion. So, that is the principle behind sequencing. So, when a nucleotide is incorporated, one hydrogen ion is released, and because of that release, you get a change in the pH, and that change is recorded by the machine, and that's how you get a base call. Like what kind of base is this? Because you know that which nucleotide is entering, and if a nucleotide is entering particularly and you have a hydrogen ion or change in the pH, so you know that this nucleotide has been incorporated. So, if there is no hydrogen ion, so it means that nucleotide is not present at that particular position. And sometimes you can get a change in pH, there are two hydrogen ions are released. It means there are consecutively two nucleotides, same two nucleotides. So, these are all next generation sequencing. Means earlier, when these platforms came, there was a generalized name, next generation sequencing method. But beyond that, then further innovation came. Another type of sequencing methods emerged. So then they became second generation sequencing method, because now the new methods, they were called as a third generation sequencing. So in nutshell, don't get confused. Next generation sequencing method is a common terminology beyond Sanger, right? And then within that, you have second generation sequencing methods, whatever I have talked about till now. Then third generation sequencing methods have come. So there are two major methods which I will talk. This is one, PacBio, single molecule real time sequencing, SMRT based method. So there are no PCR steps. So here actually the DNA fragments, they are sequenced in real time. So there again, there are different platforms. So this all these pictures are taken from their respective websites, and there may have been further updates also. These may be around few months old, but because these technologies are so fast evolving, even analysis methods are also so fast evolving. You can have a new method maybe coming up tomorrow. So this is the most latest as of now, which I know about, the sequel 2E. And again, here there are certain stats that what kind of data they can generate and how much data they can generate. So the number of reads, it can be around 4 million in 30 hours. So now you can say that 30 million is not too much as compared to if we talk about Illumina platform, where the output is 16 terabyte also up to. But the beauty of these methods are, these are long read. Their read length was just 150. 150 from here, 150 from here. That's it, in the paired end mode. But here the read length can go on an average 20 kilobits. Of course, there may be smaller fragments also, there may be larger fragments, but on an average, a single sequence can be 20 kilobase also. So that is the beauty of these platforms. This is another platform. Again, qualifies to be a third generation sequencing method. It's a Oxford nanopore method. You can see it looks like a stapler kind of machine. This is one of the variants with the low throughput, MinION, it is called. So this is how actually this method works. You know that there is a membrane. So the DNA goes through the pore on the membrane. So it's a nanopore. That's why it is a nanopore. Method is also called nanopore. At the top of this pore, there is a topoisomerase enzyme. What topoisomerase does? It unwinds the DNA. So you can see that only one fragment is passing through it. And when this is passing through it, there is a change in the current for each nucleotide. And if it is ATGC, accordingly, there will be a differential change in the current, and that change in the current is recorded by them. And that's how the data is read. Again, it is a long read technology. There are so many variants for this, different throughput. You can see throughput, it is going from 50 GB to 250 GB, even when you can run 48 such cassettes together. So output could be 14 terabytes in one run. And the runtime is 3 days for this. So this first version MinION, even we have run it within the lab. So when this technology came, the major advantage of this was you don't need a very kind of high throughput machine where you need to invest. This actually used to come along with the consumables, the reagents, how you prepare the library and load it. So it used to come as a free. I don't know what is the situation nowadays. So it's a stapler kind of machine, and later on actually they came up with this version. It is not mentioned here. So they came up with the idea that sequencing while on move, like you load your machine, you put it on your side seat and you can drive also. They added some computational infrastructure, means a computer related hard disk and processor, those kind of things, that data will, sequencing is happening at the same time it is being recorded in the hardware of the machine. So sequencing while driving, so that kind of thing they came up. I don't know right now that machine is still available or not. So Mini something they named it. So now this is a very snapshot overview of this. This may be a little bit old. So but I can I just want you to majorly focus on three methods which are prevailing in the market nowadays: Illumina, PacBio, and Nanopore. So Illumina is a short read platform, so where your read length can be 150 or 300 nucleotides, and in the long read, PacBio and Nanopore, you can generate the sequences up to 20 kilobase on an average. Even in Nanopore, sometimes they claim that the sequencing read can go up to 1 megabase also. We have done in our lab ourselves 200 kb or 250 kb we also were able to achieve in a single read length. But little bit issue with these two platforms, what I have observed myself is the quality of the data. PacBio and Nanopore means initially they launched, their error rate was little higher. Is 85% accuracy was there. But now with the evolution in their chemistry, their methods, now they also claim that it is more than 99%. And by increasing the sequencing depth, when I'm saying sequencing depth, it means like told you that when you are sequencing a genome, at least 10 times you need to sequence. But when we do the next generation sequencing, we talk about 50 times or 100 times. So if you are sequencing it multiple times, you can reduce the error rate. How? Because every time you don't think that there will be, or it is the possibility of having an error at the same position is not there. Maybe in one read there is an error, but at the same position, if you are sequencing it say 50 times or 100 times, you may not have an error. So that is why you do the sequencing multiple times. So with the higher sequencing depth, maybe your error rate will be down. So this is again nanopore platform is not here. So if you compare different platforms with respect to the read length, which is on the x-axis, and your total data output, which is on the y-axis. So you see this, where is the Sanger? So in terms of read length, it's reasonable around 1 kb. But if you go with the depth of sequencing output, so it's the lowest. All others are next generation sequencing methods or different kinds of platforms. You see the PacBio with the largest read length. If you add nanopore, it will also be there somewhere. Again, this is little older because their throughput has also increased, so they can go even higher up to that level. And here again in Illumina also NovaSeq has not been added. The throughput is now again further has increased. So this is just to give you that where actually these different platforms they stand in terms of different features. Now this is what I just told you. So the base accuracy for NGS platform is 99.9%. Although they claim 99.999% also. So one error every 1000 base, which is still lower than the Sanger. But if you can sequence it with high read depth, so those errors can be taken care of because you are sequencing the same fragment multiple times. So when you do the sequencing, how the output looks like. You have heard about the FASTA format, file formats. So where the first line starts with greater than sign, and then you have some description and water here. So then in the second line you have actually your DNA sequence. But in the FASTQ it is a 4-line format. And it's a very standard format for any NGS output and downstream data analysis. So look at the 8 lines here. So where actually the sequence has means 2 different reads are written. So the first line is again kind of the description, but it starts with the @ sign, not greater than sign, right? Then the second line is your actual sequence. Third line starts with the plus sign. So these are very important. These are not just signs. If you use these files in any downstream analysis, if you miss even one single @ sign or plus sign, your file will not be read by the tool. It will be like shown as corrupted or not in correct format. So third line starts with the plus sign and again the exactly same description which is there in the first line. And the fourth line actually gives the quality of each base of nucleotides which are there in the second line. So what is the difference in FASTA and FASTQ? FASTA 2-line, this is 4-line format, and here you have the quality information also. In FASTA you don't have the quality information. And you can put millions of the sequences in a single file. Actually, in the first line, when you have lot of these things, you can see when the output comes from the machine, so it has the information of, means it's a very unique ID. It has the information of what machine you have used, in that machine what flow cell you have used, what is the lot number of that flow cell, and within that flow cell at what position. You can see those flow cells are shown. At what position this particular read has been sequenced. Means you can trace it back. Anybody can trace it back. Actually, even because if you know that which machine has given an output, and every machine has a unique ID, so you can even find out where, which lab this has been sequenced. But generally, when you get the output, the people, means the company people, they modify it. Means one thing is you don't want to give it a tracking as a user also. Other thing is that you, because if you have so many things written here, your file size will be increasing. Because here we are not talking about 10 or 100 sequences. We are talking about billions of sequences in a single file. So sometimes our file size is just single file is 100 GB, 200 GB, right? And you want to decrease the size of the file as much as possible. So maybe they will give the unique ID there, then the sequence, of course. In the third line, they just put the plus sign. Even they don't give ID. So it is assumed that it is exactly same which is there in the first line. And the fourth line is, of course, again very important because it gives the quality of the sequence. So this is a very, again, important point, that how the output looks like in any NGS data file. Thank you. So this is the Fred quality what I talked about. So it is in the form of the probability, how the Fred score is calculated. This is called as Q. And 10 Fred score, it is 1 by 10, 90% accuracy, 20 is equal to 1 by 100, 99% accuracy, 30 is 1 by 1000, 99.9% accuracy, 40 is 1 by 10,000, 99.99%, and 50 is 1 by 100,000, which is 99.999% accuracy. And in the FastQ format, actually your Fred scores are not from 0 to 40, which is the standard in a Sanger format. So they are always in the Fred plus 33 encoding. Means always there, it starts from, as you can see, the very first line. The first nucleotide starts from 33 and it goes to 126. So 33 is actually equivalent to 0 of the Fred score, which we used to talk about in Sanger. I am not going into this detail, so otherwise it will be too technical. Now, once you have the sequencing data generated from any platform, very important aspect is data QC. When you have less, you need, again, data QC is required, but maybe you can expect that there will be very good quality. But when you have your so much high throughput data, a lot of data, it becomes a must thing, because your downstream analysis will be as good as your data. So there are a lot of tools available for this. It's mentioned only one tool here, FastQC. So there could be different kinds of things here. One major point is the quality of the read in terms of the Fred score. Because although different companies or different platforms, when you reach out to any company for providing these kind of services, they can say that बहुत अच्छा output है, will give this, but it may not be. How to ensure it? So you need to do that segmentation, followed by adapter addition, and followed by size selection. You select a particular size. If your library is not good, or even if you are reaching out to the service providers, they have not done a good job, so there could be contamination of adapters also. Maybe they have sequenced a smaller fragment. So you are expecting 150 long, base pair long read, but maybe their initial fragment size was 100 only. So other was adapter. So when you will do the sequencing in your data, again adapters will come. So you need to identify if there is a adapter contamination or primer contamination, because that is not the actual sequence what you are sequencing. So those needs to be identified, and if it is there, it needs to be trimmed also. So you need to delete those bases. You need to remove those bases from there. So this is another aspect of this. Another aspect could be, as I told, that you can pool several samples. What if there is some contamination has come from some other sample? It is again a lot of human manual thing also. Somebody is segregating those data. Even if somebody is preparing the library, they are adding some barcode, and they are pooling the libraries. What if someone who is working or is preparing the library added a wrong barcode, or same barcode to two different samples coming out from the two different labs? So those also needs to be checked. So if your data has some read contamination from some other, means if it is from the same organism, maybe you will not be able to identify. But for example, if I am working on rice, and if I have a contamination of human data, so I can identify, because I can map those or align those against the human genome and can see that yes, these are the reads which are not coming from rice, but they are coming from the human. So it means there is some problem with the data. So I am just mentioning 2-3 examples that what kind of problems could be there for the data sets, and QC is very important part before you start with the analysis. Now I will give you some idea about what all kind of things you can do using NGS methods in terms of applications. And I'm sure if you have heard, so already it is a part of now some of the clinical investigations. I'll give some examples if the time will be there. It's just a cartoon that with it means the time will come, so this will become a kind of menu. Like you go to Lal Path lab or any other diagnostic lab, they'll ask what kind of tests you want to get it done. So it will also come into a standard menu that what you want to what kind of sequencing you want to get it done. So these are some of the applications listing. So I will take some time to explain some of these at least, which are important in any routine molecular biology or genomics kind of experiment. So very first is the whole genome. At the genome level, de novo sequencing means like human genome we are talking. Maybe you are working on some new organism, new species, and you want to know how its genome, what kind of arrangements of ATGCs are there, what genes are present there, what all other than genes also what is there in its genome. So you can do the whole genome sequencing, and the speed you have already seen. Only the part of the sequencing. Again, analysis is manual kind of, means you have a lot of tools available, but you need your own brain. Means some scientist should be there to understand it, what the data is, or even if you are using several tools which are available, the output, is it worth which can answer your question or not. There are several tools which can do the assembly part. Again, is related to the whole genome sequencing, because as I told, even you do the Sanger sequencing, what is the read length? 1000 bases. You do the short read sequencing using Illumina, 150 bases. You do the nanopore or PacBio sequencing, average length is 20 kilobase. But your genome is 3 GB. 3 GB is in 23 chromosomes, right? So you expect only 23 fragments for 3 GB. But your data is so fragmented, 1 KB fragments, 150 base pair fragment. So you need to put it together again, so that you can construct this whole chromosome and the whole genome. So in the very basic view, how you reconstruct is you have one read, you have another read if there is overlap. So this becomes from here to here. So this way it goes. But there are several algorithms which I will not talk about. But this is in very layman term that you find the overlaps between the reads and you keep on extending it and you make the whole chromosome or the whole genome. But there are a lot of complexities in the genome. Lot of repetitive sequences are present. Sometimes you have had transposons, transposable elements. Like to this, there are several other kinds of repetitive elements also. And sometimes these repetitive sequences can be in KBs as well, kilobases. So now if there is a repetitive sequence of say 1 KB and your read length is 150, how you are going to resolve? Repetitive sequence means same sequence is present here in the genome and same sequence is present here also. Now you have a 150 nucleotide long read, you cannot make out that this is coming from here or this is coming from here. Right. So there are again several methods, several algorithms have been developed to resolve it up to some extent. Not 100%. Again, you need brains behind those. And even after putting brains, maybe you will not be able to resolve it 100%. Again, the power of technology comes. This human genome was first reported in 2003. I think 2 years back we had a paper on human genome when actually the human genome was completely sequenced. Because at that time there were a lot of gaps because they had one fragment, but some part was missing in between. There were certain repeats which were unresolved, but now they have been able to do it maybe after 20 years or so. Right. So there are a lot of things, and here actually now the job of us like you that comes because sequencing has become a commodity. You isolate the DNA, you send it to a company, they will sequence it, right? But the actual job starts when the data comes, how you analyze that data, and this is for that purpose. There are certain applications which are mentioned here. So de novo meaning we do not know in the sequence of that particular organism as of now. We we are just doing it for the first time in blind. Then resequencing. You already have a human genome sequence available, but I want to know what is the sequence of my genome, or you want to know what is the sequence of your genome, or any particular individual, or a particular patient. Because there are several diseases which are associated with the DNA sequence, genetic disorders and many others also. So we want to know what is the change, what is the difference in the nucleotide sequence that may be causing a particular disease. So this is one example I am giving. So there are many applications around this resequencing. So this is the concept behind resequencing, means you already have a standard genome available, but you are sequencing many individuals. In plants, if I give you example, when you go to the market, you see different kinds of rice, maybe 20, 30, 40 different kinds of rice. Ultimately, it is rice, but it is the difference in the fragrance or how it looks like, size, thickness, all those kind of things. So there must be some difference in their genome, their DNA sequence, which makes it or which gives it a particular kind of characteristic. So all those you can do via resequencing. Within resequencing, another related aspect is targeted resequencing. When we say only resequencing, meaning you are sequencing the whole genome. But targeted meaning rather than the whole genome, you are just sequencing some specific regions of the genome. And very important or good example is, which is now in clinical aspects also, in diagnostic labs also it is available, like cancer panel. So already people have identified, say, like these are the 10 genes or 20 genes, and within those genes also there are certain changes in the nucleotide sequence which are associated with a particular type of cancer. How you can detect it? So there are assays that they will do a targeted sequencing only for those 10 regions rather than whole genome. So this will lower down your cost. You are not sequencing the whole genome, only maybe some 10 regions. So within the same machine or same place, you can sequence many more samples. So you can lower down your cost. So, and actually that is, as I give the example. So if you go to some standard labs, or many startups have come up with those kind of panels, cancer panel, what they call. This is one example, but like Alzheimer's or schizophrenia, for these kind of, which are very standard genetic kind of diseases. So there are certain panels available, and lot of research is still going on, because sometimes happens there is a cancer patient in US and they have identified this kind of panel, but it may not work in Indian population, because nowadays it is not only the DNA sequence, it is your environment also. So earlier we used to talk about only G, now it is G into E. There may be a different mutation here or in a particular population that is associated with the cancer. So there are several projects actually going on in this direction. So some are sequencing 10,000 individuals, some are sequencing 1 lakh individuals, some people they have even 1 million project also, and they are trying to find out these kind of mutations which might be associated with certain diseases. It's a very important application of NGS. The mutation detection is also related to it. Amplicon sequencing is also related to it. Again, Amplicon cancer panel. So this is all what I have covered in this. So these are only few applications I'm telling. So a lot of innovation is happening. You can read any journals. Nowadays you will find some one or more paper which is associated with some new application which is NGS, related to NGS. Again, you have to see, sequencing part remains the same. It is how you prepare the DNA for sequencing. So that there it is the innovation. So that DNA, how you are, or RNA you are getting it and to which it is associated with, that defines your application. So same thing is at RNA level also you can do. So maybe I am not interested in whole genome. I am only interested in what are the genes which are expressed. So rather than whole genome, I can sequence only the RNA. So it can tell that what genes are expressed. Or even if sometimes, if I have the whole genome, I have some annotated genes also. But you know, RNA is dynamic. Genome is everywhere, it is same in all the cells. But RNA can change. If I am standing here in AC, when I go to outside 45-degree temperature, some of the genes, their expression will change. So it's very dynamic and very much specific to the biological context. Again, lot of projects around this. You want to study that how a gene function or how the regulation of a gene changes at the RNA level. So you want to identify what is expression level. So this is again at the whole genome level. All the genes you can monitor at a single point of time. Same thing you can do at the regulation level. Small RNA sequencing you can do. MicroRNAs you must have heard. They are regulatory molecules. You can do the sequencing of those. Again, even at the epigenetics level, you can do a lot. You know, there is a transcription factor which binds at certain positions in the genome. Then it regulates the expression. So now if you want to identify where a transcription factor is binding. So if you can, again, if you can find out that stretch where that protein is binding, you can sequence that. You can find out that which is the binding site. So I don't know if you have heard chromatin immunoprecipitation. It's a method. So again, this is how you are isolating that DNA. So using chip chromatin immunoprecipitation, you find out the reason. You actually you are precipitating a protein, and along with the protein, you are precipitating the DNA where it is bound. If you can separate out that DNA, you can sequence it again, and you can find where your protein is binding. So these are some of the applications. So like this, there are so many applications. This is what is there in all these slides. Again, just one more aspect I want to explain. I just mentioned that from one end, two end. Sometimes I mentioned 2 into 150 was written, 2 into 300 was written. So this is because of the sequencing can happen in two modes. One is the single end sequencing. If there is a fragment, you sequence only from one side. Paired end sequencing, you sequence it from both the sides. So that is paired end sequencing. Another concept was there, mate pair sequencing. Nowadays it is not used. Earlier for Illumina, long read libraries were being formed. Means rather than smaller fragments, 20 kb fragment you will clone and then you sequence it from both the ends. So there was a special method for preparing a library and sequencing it, but nowadays it is not used. Barcoding I have already explained. This is barcoding. When you have multiple samples, you sequence them individually, so there is no barcode. If you have multiple samples, you prepare individual libraries, adding the barcode, and if you sequence it in the same physical space, you can see that the dots of different colors are there on the right hand side. And then later on, based on that color, means barcode, the unique nucleotide sequence, you can separate it out the data, and that can lower down your post. So this is again some cartoon that how the complexity of the analysis increases in the NGS. It's something like Sanger. You have only few data points. So you can easily connect it and you can make what you want. But in NGS you have millions of data points. Now it will depend on you how you want to. There could be several possibilities how you connect it. You can make it like this. You can make it like this also. So what basically this cartoon or this slide shows that there is a lot of intervention which is needed from the scientist or the researcher side, and you have to decide the right way, right tools, right algorithms, and then once you have the output also, you need a way to validate that yes, this is what was expected. So this is one of the forum if you are interested in this area, sec answers. If you are actually doing some data analysis, you can register here and you will find answer to a lot of queries, because this is the forum that means people give your questions and you get kind of immediate answer. And with my experience also, 90% of the time there is already an answer with my queries, because already this community has grown a lot. But even if it is not there, so somebody will be there that who can reply to you. So these are some considerations when you do any NGS experiment. First thing is it's a very fancy thing, but we need to understand, is it really required for our experiment? Many people just do it by name, being fancy. So then what kind of applications I can do, or what application I need to do? Then for that particular application, which technology is the best? What is your experiment design? Again, sequencing is just sequencing. That you can control the quality of sequencing. Rest is all behind, we are there. How we design the experiment, how we handle the data, so it depends on us. Do you have the expertise for the bioinformatics, means data analysis part? Then even if you do the analysis, can you find a way that you can judge whatever you have done the analysis or output, is it correct or not? Do you have proper statistical algorithms or some statistics which you can use to evaluate? So one very important is rubbish in and rubbish out. So this all depends on what is going as an input. If we just talk about the sequencing technology, so the quality of the data should be very good. Means you have designed a very good experiment, you are very good in data analysis, but something is wrong with the data itself. You can't do anything, right? So if we just talk about the technology, so it's very important to have a very good quality of the data. So whatever technology you have, the data analysis is the bottleneck. So this is a sum up slide. Sequencing is a commodity, but it can be outsourced very easily. bioinformatics or data analysis is the essential point of science, and data analysis and discovery of meaning in raw results. As the data throughput increases, the cost and time spent on analysis it increases linearly, and these platforms are evolving very rapidly. New technologies, new file formats, new data types, and different platforms they have different standards. Sometimes it becomes difficult to get the data on a single platform if you want to club the data from different platforms. But nowadays, quite a few tools are available, but we have to use them very wisely. Very rapid evolution of methods, very rapid release of methods and softwares, mostly are unsupported, means because they are public. So if there is no obligation that somebody who has developed, so they are obligated to help you. But most of the time, you will see scientific community that you will get a reply. So large data size, so it is always ultra high throughput plus lower cost. So it leads to broader applications, and this means you can do a lot of innovations and more studies. But you need a better infrastructure, more computational power, and beyond this, more manpower and skills. That's where generally I stop. That there's a lot of potential in this direction, but yes, you need to develop those skills, and it's very important and not very readily available. I'll say it's available, but not very readily available because you have to think both biology as well as computational part for doing this kind of data analysis. With this, thank you very much. I don't know. I hope some of this you have been able to absorb, but at least my idea is that you should know about what kind of methods are available, what is the power, where this kind of science is going on, and you can watch some of the videos at least today if you go, so something will remain fresh in your mind. And then whenever there is a required, so at least in requirement of your research, at least you will know we have heard something or we watch some videos, so is it possible, so let us explore more. That is the idea behind this. Thank you very much, and please ask any questions. No questions? Any kind of questions? Don't feel like, maybe sometimes as a student we are hesitant that पता नहीं क्वेश्चन ठीक भी है नहीं है. There is no standard of any question, right? Please ask. Here, what we are is encoded in our DNA. If it is in their DNA, you can find it out using NGS methods. If something is beyond DNA, you may not. Barcoding here, it just means that you are adding a specific nucleotide sequence to individual samples so that you can pool and you can do the sequencing all together and you can cut down your cost. So this barcoding is only that. So say my requirement of the data is only 500 GB. But you are using a machine which can give you an output of 1 terabyte. Why you will run a machine or full flow cell of 1 terabyte when you just need 500 GB of the data, right? So this will be wastage. Maybe you will be spending 20 lakhs for that run, but your requirement is very less. So what, as a company or as a user of the machine, I will do it, I will wait for some samples from him, from somebody else, When I will have sufficient number of samples, that total requirement is 1 TB, I will prepare the library using the barcodes, I will pool them, and then I will do the sequencing. That's what the company people, they do. So that is where the barcoding comes into the picture to lower down your cost. Okay. Any other query? Feeling hungry, right? Yes. Platform. Platform, I mean to say here technology. So like when I'm saying Illumina, so it is a platform. It's a Illumina platform. So as such, platform has no meaning here, right? So platform is something, means I am giving a name, or it is just used to define a technology. So Illumina is a platform, PacBio is a platform, Oxford Nanopore is a platform, or you can say Illumina technology, PacBio technology, or Oxford Nanopore technology, right? PacBio. Short read is Illumina. PacBio and Nanopore, they qualifies to be long read platforms, long read technologies. Because in short read, you are getting individual read is 150 or maximum 300 nucleotides. But in long read, the average read length can go up to 20 kilobase. Right? Any other query? None? Okay, thank you and all the best.